This is the Chapter 15 lecture. It is the second part of our host defense, which we started in Chapter 14. So to remind you where we've been, in Chapter 14 we discussed innate immunity, which is comprised of the first line of defense. The goal of the first line is to prevent invasion by potential pathogens in the host. The second line of defense is going to be the part of innate immunity that responds to invasion. So if a pathogen gets past the first line, the second line of defense will then deal with it initially. The response of the second line is going to be nonspecific and the same for all types of microbes. In Chapter 15, we're going to discuss acquired or adaptive immunity, which is comprised of the third line of defense. And the difference in acquired immunity is that it's a specific response to invasion, so each microbe is responded to in a specific fashion. It's going to be slower than innate immunity, and it's going to retain memory of a pathogen. Some definitions we're going to use a lot in Chapter 15 are antibody. These are the proteins made by B cells, which are part of the third line of defense, and they're going to be proteins that are charged with recognizing and binding to a specific antigen. An antigen is any substance that causes the production of antibodies. They're so named because they are antibody generators. They can be microbial or non-microbial. So in our course, we're going to focus on microbial antigens. And a specific region of an antigen that is recognized by an antibody is called an epitope. So in this image, we have a bacteria cell, and it has different molecules which are drawn in a simplified way with triangles, squares, and then rounded squares. And we're showing the production of two types of antibodies, antibody A and antibody B, which have both been made by the third line of defense. And these antibodies are going to bind to specific parts of the bacteria. These specific parts are called antigens. And the part of the antigen where the antibody interacts is the epitope. The acquired or adaptive immune system, or the third line of defense, is made up of T cells and B cells. The T cells are going to bring a cell-mediated immunity, and the B cells are going to bring the ability to produce antibodies, which is also called humoral immunity. The development of B and T cells occurs in the lymphoid tissues, so these are tissues of the lymphatic system. Because B and T cells are white blood cells, they're called B and T lymphocytes. So on the left part of this figure, we see the development of a B cell, which starts as a stem cell in the bone marrow. And that stem cell lineage turns into white blood cells, which will then differentiate further in the bone marrow into B cells. The B cells will then undergo testing to ensure they work correctly, and then they'll travel to the lymphatic system. A small number of them will circulate in the host, and the majority of them will be present in the lymph nodes. The right side of the figure shows the progression and development of T cells or T lymphocytes. So like the B cells, they originate from stem cells in the bone marrow, which then turn into a general form of white blood cell that migrates to the thymus, where it further differentiates into a T cell. And like the B cells, the T cells will then be tested, and then they'll be released into the lymphatic system, most of them residing in the lymph node. There are several things on the surfaces of B and T cells which will be important to us in our descriptions in Chapter 15. So the first thing I'd like to point out to you is the presence of MHC2 markers. So these are short for major histocompatibility type 2 markers. And this is a receptor on the surface of the B or T cell, so it's protruding from the plasma membrane of these cells. And the MHC2 marker is going to be very important for presenting or binding to antigens and showing them to other immune cells. Not shown in this figure, but that would also be present on the surface of B and T cells, is the major histocompatibility type 1 marker, or MHC1. So MHC1 is similar to MHC2 in that it protrudes from the surface of a cell, but MHC1 is going to be present on all human cells with a nucleus. The only type that don't have a nucleus are red blood cells. So red blood cells are an exception, but all other human cells have an MHC1 marker, B and T cells included. And the purpose of the MHC1 marker is to identify the cell as part of the host, which we refer to as self. So this is how the immune system distinguishes self, or cells that are part of the host, from non-self, or antigenic microbes or other invading substances. Okay, on the surface of the B cell on the left, you'll see labeled the B cell receptor. Looks like a Y. This B cell receptor is going to be unique on every B cell. So there's a lot of different combinations of B cell receptor. And each B cell receptor will bind to one antigen. So the area of the B cell receptor that interacts with antigen is colored in yellow. On the surface of the T cell, you see the green squiggly looking receptor, which is the T cell receptor. And like the B cell receptor, this will be unique on every T cell and will interact with one specific antigen. Also shown on the surface of the T cell are molecules CD3 and CD4 or CD8. We're going to focus on CD4 and CD8, not discuss CD3 any further. So CD4 is the receptor that will be present on T cells, which are categorized as helper T cells. And CD8 will be present on the surface of T cells that are categorized as cytotoxic T cells. And we'll talk more about these CD4 helper T cells and CD8 cytotoxic T cells in a moment. Okay, so when B and T cells are created, so these B and T lymphocytes have to undergo testing, first to ensure that they do not interact with self antigens, and second to see if they bind to foreign antigens. So at the top of this figure we have 8 lymphocytes shown. So these could be 8 B cells or 8 T cells. And each of these lymphocytes has a unique B cell receptor or T cell receptor on its surface. So what's being shown at the top is these B and T lymphocytes are being exposed to human cells, which are labeled self. And ideally, the B or T cell should not interact with self. You can see that 2 of the lymphocytes have B cell receptor or T cell receptor that is binding to some molecule on the human self cell. So these 2 lymphocytes are eliminated, which is indicated with the red X. And by eliminated, they are activated to undergo apoptosis, which is also called cell suicide. So essentially, they destroy themselves. The reason they have to be destroyed is if the immune system responds to self, then it will attack the host's own cells and tissues and try to destroy them, thinking that they are an invading microbe when they are not. So these 2 cells that respond to self have to be eliminated by apoptosis, and so they are killed in this early stage of testing for the lymphocyte. So this is how the idea of immune tolerance is established, which means the immune system does not react to self, but it does react to foreign invading molecules. Okay, the bottom of the figure is showing now all of the lymphocytes that passed the first test. So these were all not reactive to self. So we have 6 left. Now each of these 6 types of lymphocytes will be allowed to circulate in the host. And when one of them interacts with an antigen, which in this figure is shown as small red triangles, when it binds to the antigen, that lymphocyte will be activated to replicate. So then you have a whole population of clones, which are identical to the original lymphocyte that bound to the antigen. So in this example, we have the lymphocyte with the red B cell receptor or T cell receptor, depending on what type of cell it is able to bind to this particular antigen with its receptor, and that activates that lymphocyte to replicate so that now there is an entire population of lymphocytes identical to the original that can all bind to the antigen invading the host. So here we have the major histocompatibility complexes. So on the left we have MHC type 1. On the right we have MHC type 2. So we said earlier that MHC type 1 is going to be present on the surface of all self cells, including B and T cells. And MHC type 2 is more specific. So it will be present only on the surface of B cells, T cells, and other cells that are antigen-presenting cells. So the purpose of these MHC molecules is to identify cells as self, and then also to show antigen to other immune cells. So it's a way they communicate with each other. By binding an antigen onto MHC and displaying it on the surface, they can show other immune cells what antigen is invading the host. The types of antigens are threefold. We have extracellular pathogens, which are going to be molecules that are on the outside of microbes, and these are microbes that are outside of human cells. So the microbe has invaded the host, it's inside the human, but it has not gotten into a human cell. So it's freely floating in the human. So all the molecules on its surface are going to be antigens, and they will be responded to in a particular way. The second group are the intracellular pathogens, which are also called endogenous antigens. And these are microbes that have invaded the host, so they've gotten into the human. They have also further invaded one of the host cells. So shown in the second image is a human cell, which has in its interior a virus. So this intracellular virus is the endogenous antigen. So that has to be dealt with in a different way than the exogenous antigen shown in the first image. And then the last category are the self-antigens. So we already saw this in lymphocyte testing a couple of slides ago. So self-antigens is any molecule on the surface of a human cell. And so these ideally would not be responded to by the immune system. But in some disease states like autoimmune diseases, the immune system loses immune tolerance and does respond to self-antigens incorrectly and attempts to destroy cells of the host. So the cell types that are responsible for dealing with the exogenous antigens in the host, so that was the first group in the last slide, are called antigen-presenting cells, or APCs. So these cells are capable of presenting exogenous antigens to other immune cells. They also are capable of presenting those antigens on MHC type 2. So MHC type 2 is going to be present on all of these antigen-presenting cells. We also said it was present on T cells, and that's the only cells in the human that have MHC type 2. So the groups of antigen-presenting cells are macrophages, dendrites, and B cells. Okay, so this is what happens when an antigen-presenting cell encounters an exogenous antigen, so what it does after that point. So shown in purple is an antigen-presenting cell, which encounters a freely floating microbe in the host. So because the microbe is freely floating, it is an exogenous antigen. So the antigen-presenting cell will adhere to the microbe, probably via pattern recognition receptors, so general patterns that are seen in foreign molecules. The antigen-presenting cell will use those to recognize the microbe as a microbe and something that has to be destroyed. It will then take the microbe into its interior, It will process the microbe, meaning it will break the microbe up into individual molecules. It will then bind one of those molecules that derives from the microbe to its MHC type 2, and then it will put its MHC type 2 bound to the molecule derived from the microbe onto its surface. So shown in the zoomed in box is the surface of the antigen presenting cell, and it shows its MHC type 2 receptor, which is bound to a molecule from the microbe. So that little pink figure 8 is a molecule that originates from the microbe. So this is the presentation part of the figure. So if an exogenous antigen is encountered by an antigen presenting cell, the antigen presenting cell will process the antigen and then present part of it on its surface on MHC2. The purpose for doing that is illustrated in this figure. So this is at the top, we have a antigen presenting cell, which happens to be a B cell. We have a tiny microbe that is an exogenous antigen because it's freely floating in the host. So in step 2, we have the antigen has been processed by the antigen presenting cell and is now being presented on the surface of the antigen presenting cell. It's being presented on MHC2. And when it presents on MHC2, the antigen bound to MHC2 will be recognized by a T helper cell, which is abbreviated TH here. And it's a T helper cell because it expresses CD4 on its surface and because it can recognize antigen presented on MHC2. So the interaction between the antigen presenting cell, which happens to be a B cell, MHC2, the processed antigen, and the CD4 expressing T helper cell is shown in the zoomed in box. After the T helper cell is presented antigen, the T helper cell will then activate various immune cells to respond to the antigen. So what happens in this figure is the T helper cell activates the B cell that's presenting its antigen on MHC2, and the B cell can be activated to either become a regulatory B cell, which will make sure the immune response doesn't get out of hand. The B cell could be activated to become a memory cell, which will have a long life, will circulate in the host, and will be ready if the antigen infects the host again. So if the same microbe infects the host many months, years, or decades later, the memory B cell will be ready to respond. Or the T helper cell activates the B cell to become a plasma cell. And the job of the plasma cell is to simply make antibodies. The antibodies that the plasma cell makes will be identical to the B cell receptor that was on the surface of the B cell. They will just be released to circulate in the host. And shown in the bottom right corner of this figure are antibodies, which look like red snowflakes, binding up to the microbe, and that will target the microbe to be destroyed. So this figure summarizes the function of antibodies. So these are the proteins made from a B cell that's been activated to become a plasma cell by a T helper cell. So antibodies can do many things. The first thing they can do is opsonize, which means they coat the microbe. So in this image, the microbe is a pink-looking bacillus, so it's oblong in shape. And in the opsonization illustration, the microbe is coated in antibody, which will enhance the phagocyte being able to destroy it. So the phagocyte is shown in purple. Antibodies can also neutralize microbes, which is shown in this virus being coated in antibody, so it can no longer bind to host cells and get into them to infect. Antibodies can also agglutinate, which means bind to several microbes at once and gather them into a specific area. or antibodies can activate complement, which you may recall from chapter 14. If an antibody binds to antigen, it activates the classical complement pathway. So the outcome of that activation in this case is cytolysis, where we have a membrane attack complex punching holes in the surface of the microbe, and the microbe will be lysed as a result. And then lastly, if a microbe is damaging the host by releasing a toxin, the antibody can bind to the toxin and neutralize it so that it cannot cause any further damage to the host. There are 5 types of antibodies. They're all made of the same kind of protein called immunoglobulin, which is abbreviated Ig. So we have immunoglobulin G, immunoglobulin A, immunoglobulin M, immunoglobulin D, and immunoglobulin E. So the way that these antibodies look is slightly different. Some of them are made up of multiple subunits. Some just have one Y-shaped protein. You won't be tested on the structure of them. I would like you to know that the immunoglobulin G comprises 80% of the antibodies in the blood serum, so that one is the most prevalent. I would also like you to know that IgM is the first type of antibody made in response to an infection, and that IgA is an antibody that we secrete from our mucous membranes. So every time you blow your nose, there's a little bit of IgA in the snot. And that is all summarized in this slide with text.